Welcome to our module on preventing GMO contamination.
This module was authored by Adria Fernandez, Jim Riddle, Christine Moncada, Constance Carlson, and Craig Schieffer in 2017 and updated in 2026. Narrated by Constance Carlson, with additional narration generated using the AI voice of Hannah from 11 Labs.
First, we'll talk a little bit about what is a GMO, GMOs and organic, how contamination occurs, what the consequences are, how to protect yourself, and we'll go over the current status of GMO crops.
Here we show a picture of corn, a commonly genetically modified crop.
GMO stands for genetically modified organism. The term can be defined in different ways. Most simply, a GMO is an organism, often a plant, but it can also apply to animals, bacteria, or fungi, that has been bred using methods that directly alters the genetic material using genetic engineering techniques. The USDA's National Organic Program provides a specific list of breeding techniques that are not permitted under organic standards. Genetic engineering is used as a synonym for genetic modification, so you will see the terms genetically engineered and genetically modified used interchangeably. Another term that is sometimes used interchangeably is transgenic. Transgenic is a type of genetic modification that means the DNA that was transferred came from an organism of a different species. By extension, transgenes are the inserted genes that came from another species.
In this image, we show a simple diagram of a gene moving from the genome of one species to a new species.
We will discuss some of the specific genes from other species that are inserted into crops later. The insertion of a transgene into an organism is considered a patentable invention, and therefore the use of transgenic crops can be restricted by terms dictated by the patent holder to ensure that no one can grow crops with the engineered trait unless they pay a licensing fee to the patent holder. A trait can mean any characteristic of an organism, but in the context of genetic engineering, trait generally refers to the specific characteristic arising from the transgene. In the crop plants that we will be discussing, the traits involved are generally herbicide resistance or production of the insecticide Bt.
In this image is a soybean plant. Soybean is an example of a crop that can have the Bt trait.
Not all crops have been genetically modified. The most widespread GMO crops are corn and soybean, but there are also commercially produced varieties of GMO alfalfa, sugar beet, canola, cotton, and several fruits and vegetables, including potato and papaya. There are currently no GMO varieties being marketed for small grains, dry edible beans, peas, sunflowers, common cover crop species, or most fruits and vegetables. A GMO is specifically defined and addressed under National Organic Program regulations. In order to obtain organic certification, farmers cannot grow crops or use products that were produced using excluded methods. Excluded methods consist of cell fusion, microencapsulation, macroencapsulation, and recombinant DNA technology that includes gene deletion, gene doubling, changing the position of genes, and introducing a foreign gene.
Foreign genes are also called transgenes.
Breeding using hybridization is not an excluded method, and traditional hybrid varieties are allowed in organic production.
We show a photo of two weakly growing inbred corn plants that are the parents of some adjacent tall, vigorous hybrid corn plants in a field research plot.
GMOs are prohibited in all areas of certified organic production under the definition of excluded methods. Organic farmers cannot knowingly plant GMO seed. They also cannot apply inputs that are GMO, such as genetically modified rhizobia in legume inoculants, or genetically modified Bacillus thuringiensis bacteria in biocontrol insecticides. However, there are non-GMO versions of these inputs that may be allowed in organic. To know for certain whether an input is approved or non-GMO, you can check the listings maintained by OMRI, the Organic Materials Review Institute. During the 36-month transition period prior to organic certification, producers must use organic practices, including non-GMO seeds and inputs, but they do not receive organic premiums. Some buyers offer a non-GMO premium, especially for crops such as soybean and corn. As a result, when you are transitioning new land from conventional to organic, it is important not only to avoid using GMOs yourself, but to prevent contamination from other sources, as we will discuss in the next slides. So how can GMO contamination make its way into your products? There are several avenues for this to occur: planting stock, pollen drift, volunteer plants, and equipment. The first step to avoid GMOs is to not plant contaminated seed. Contaminated seed means that your seed stock contains some percentage of seeds that are GMO, so you are unintentionally planting a GMO crop. This can happen if a supplier sells you contaminated seed, or if you replant seed from your own crop that was contaminated by pollen drift or was somehow mixed with GMO seed in storage. You can request GMO test results from seed suppliers. There is no way to visually distinguish GMO from non-GMO seed. Only a genetic test can say for certain whether transgenes are present in a seed. Pollen drift is the second and perhaps the most difficult to prevent source of contamination. Because pollen is genetic material, it can carry GMO traits that will be transferred to the plants it fertilizes. This occurs when GMO pollen from nearby fields is carried by wind or insects to your field, where it fertilizes your crop, producing grain that is a GMO hybrid. Several circumstances must be in place for GMO drift to occur. First, there has to be a GMO counterpart to the crop. Next, that GMO crop must be grown near a susceptible crop. And lastly, the crop should have a breeding system where outcrossing or cross-pollination is likely. Cross-pollination is the transfer of pollen from one plant to a different plant, either by wind or by insect pollinators.
Here we show a tassel of corn, a wind-pollinated crop, and the purple flowers of alfalfa, an insect-pollinated crop.
The organic crops with highest potential for GMO pollen drift in the upper Midwest are alfalfa, canola, sugar beet, and corn. Corn, alfalfa, sugar beet, and canola are all outcrossing crops that are pollinated by wind or insects. Soybean, because it is self-pollinated, is not generally considered to be vulnerable to GMO pollen drift. For the most part, pollen drift in alfalfa is primarily a risk in the western United States, where seed production occurs. If you are cutting alfalfa for hay before any of the plants set seed, then contamination will not be an issue. However, if even a few plants are going to seed in your field or around field margins, there is the potential for GMO-contaminated alfalfa populations to persist in your field. Sugar beet is normally harvested before flowering, so contamination is not normally a concern unless you are raising it for seed.
This picture shows a sugar beet plant in a field near harvest. The plant is vegetative and has not formed any flowers. It has been dug up from the soil to show its large fleshy root.
Canola is not widely grown in the upper Midwest, but in the areas it is grown, GMO pollen drift is definitely a concern. In the case of both canola and sugar beet, it is also possible for GMO pollen to fertilize crops or weeds of related species. This can lead to weed populations that also produce GMO pollen, which may cross back into a subsequent canola crop. Corn is generally the crop most likely to be of concern for GMO pollen drift, so most of our discussion from now on will focus on corn. But many of the same principles will apply to crops such as canola. Also, because corn is so widely grown, it's most likely to be tested for contamination and rejected by buyers if GMOs are found. To understand why GMO pollen drift can be a problem in corn, let's briefly review the steps in corn reproduction. To produce kernels, corn needs to be pollinated. The corn plant develops tassels, which produce the pollen-bearing structures called anthers. Pollen is released from the anthers.
This picture shows the tassels of corn plants with small dangling anthers that have emerged as part of the pollination process.
Pollen grains are carried largely by wind to other nearby corn plants. Pollination is when a pollen grain from the anther lands on the silk of a developing corn ear.
In this diagram, we show a close-up of a single tiny flower of a corn plant with anthers that have emerged.
Fertilization is the process where the pollen grain is transported down the silk to the ovule, from which a kernel will develop. If the ovule is not fertilized, the kernel will not form.
We show a diagram of a corn ovule with a long filamentous silk.
The developing kernel contains genetic material from both parent plants and will express traits from both. For example, a kernel that was fertilized by pollen from genetically modified Bt corn will produce the Bt toxin. Subsequently, this corn will be harvested and will test positive for GMOs. We'll talk more a little later in this presentation about how to prevent this kind of unwanted cross-pollination. Volunteer plants are those that are not intentionally planted and are harvested with your crop. GMO volunteer plants can happen if GMO seed was previously used in a field and remains in the seed bank or has been accidentally transported from a neighboring field, for example, in equipment that was not thoroughly cleaned. This accidental seed replants itself and grows alongside your crop. Some crops like alfalfa and canola can become feral, meaning that they reseed themselves and produce self-sustaining weedy populations that can persist in and around crop fields. If these populations are GMO, they can also pose a contamination risk.
In this picture, we show a soybean field with a single tall corn plant in the center. This corn plant is an example of a volunteer because it was not intentionally planted.
Equipment or storage facilities that have not been properly cleaned can also be a source of GMO contamination. GMO seed or grain can be retained or transported in equipment that has been handling GMO crops. This can be a problem when remnants of GMO grain mingle with your non-GMO crop during harvest, transport, or storage. This kind of contamination is most likely if you have a split operation growing both organic and GMO crops, or are borrowing equipment or hiring custom operators, or using shared or custom transportation to haul harvested grain. So now we've learned how contamination can occur. But what are the consequences when it does occur? Losses from GMO contamination can be a serious issue for organic farmers.
According to the U.S. Department of Agriculture Census of Agriculture 2014 Organic Survey, GMO losses have been reported by organic farmers.
According to the USDA, 92 farms reported that they lost value from their organic crops due to GMO contamination between 2011 and 2014. In Minnesota, 3 farms reported losing a total of $82,440 in value from their crops. There are likely many unreported cases. So what happens if the presence of engineered traits is detected in your organic crop? Well, if you have been following organically approved practices but end up with contamination anyway, your organic certification should not be at risk. Organic certification is process-based, meaning that you will be in compliance if you follow the correct procedures to avoid prohibited substances, including GMOs, and take appropriate measures as verified by your certifier to protect against prohibited substances in all steps of the production process. While your certification should not be at risk with inadvertent GMO contamination, you may be at risk of losing your organic market price, meaning you may have to sell your crop at conventional prices. Although there is not a uniform standard or testing requirement for organic products, some buyers may require testing to verify that GMO contamination is not present or is below a given threshold. Some foreign countries also require verification of non-GMO status for products entering their markets. If your crops contain GMO material, they may be rejected by buyers. This is, unfortunately, a problem that some growers have reported facing. So what happens if you find yourself with a contaminated crop through no fault of your own? Basically, under the current system, there is no compensation available. We may think of organic farmers as the victims in a contamination scenario, but in the U.S., there is not yet a lot of legal precedent in this area, and there is no official policy stating who is liable for lost revenues in these cases. Unlike in negligent cases of pesticide drift, where states may offer protections to victims, currently it is up to the organic farmer to protect themselves against GMO drift, and they bear the full burden of losses. So now let's talk a little bit about how you can protect yourself from GMO contamination and the associated loss of profits. Since there isn't much recourse for farmers whose crops get contaminated, the best thing you can do is protect your crop from planting to sale. The first thing you can do is to know the practices of those you do business with. Do you know your supply chain?
Your supply chain includes the seed you buy, the equipment you use, where you store your crop, and how you transport your crop.
You can do everything right during the growing season, but it won't help if you are being supplied with contaminated seed, or if your crop is commingling with GMO grain in somebody else's combine, truck, or storage facility. If you purchase certified organic seed, it should not contain GMO material. Reputable organic seed companies have rigorous controls against contamination and conduct testing of the seed they sell for GMOs. However, in transition, if you don't purchase organic seed, you should take extra precautions to prevent prohibited substances such as GMOs in seed. If you are using seed that isn't certified organic, consider looking for seed with a third-party non-GMO certification. Obtain and keep all documentation in your records. Planters and all other equipment should be clean and empty before they are used for organic crops. This includes not just your own equipment, but anyone else's that does work in your field, as well as all equipment used to transport your crop from the field or to the elevator.
This includes planters, combines, grain wagons, custom work equipment, grain bins, grain carts, semis, augers, and grain elevators.
Proper cleaning may involve sweeping out equipment, blowing out debris with compressed air, vacuuming, and running the equipment empty or purging it with a small batch of organic grain. All cleaning and storage procedures should be recorded. You should save all records of your own activities as well as documents provided by custom operators. For transport of your grain, affidavits and bills of lading should document that your crop was loaded into clean wagons, trailers, or other transport units. Make sure to keep copies of your records. You should also know your buyers and know what form or level of contamination they will tolerate. For example, for products destined for EU markets, a threshold of less than 0.9% GMO content is generally required for the product to be considered non-GMO. If you are selling on contract, know what the contract's requirements are regarding GMO rejection levels. Again, unintentional GMO presence does not mean your organic certification has to be lost, but rejection by buyers can harm the reputation of your farm and access to future sales. It's also extremely important to make sure that your organic crops aren't cross-pollinated with GMO crops, which could introduce the modified genes into your product. There are best management practices to prevent pollen drift in your fields. First, you need to know what GMO plants are nearby. Not only do you want to be aware of what your neighbors are planting and where, you want to know what might be persisting from previous plantings. With corn, it should be pretty obvious if there are volunteer plants springing up, but alfalfa and canola can sometimes persist and spread as feral populations, as weeds in the field or on field edges. Canola can also cross with wild mustard relatives, which may then carry transgenes that can cross back into related crops. Conversely, it is important for conventional neighbors to know that you are growing organic or non-GMO products. In Minnesota, do not spray signs are available from the Minnesota Department of Agriculture.
In this photo, we see a sign that reads, "Organic farm, please do not spray," which an organic farmer has installed at the edge of a field next to a road.
When you know where GMO plants are located, there are ways to keep their GMO pollen separated from your crops. This separation can be achieved either by distance or by time. First, you can use distance to keep them separate by choosing fields on your farm that are as far as possible from GMO crops of that species. This goes back to communication with your neighbors for their cropping plans. You can also plant buffers or windbreaks to provide a physical barrier to pollen drift. To a limited extent, buffers can be adjusted by increasing width or adding trees to increase height, which can reduce the flow of wind-blown pollen. Your certifier will have recommendations for your buffers based on your risks.
In this picture, we show an organic field where the vegetative buffer is a stand of pine trees.
The USDA publishes guidelines on how far plantings of GMO varieties must be to prevent gene flow out of the test fields. You are not required to maintain these distances, but you may find them a useful guide when assessing whether a neighboring field of a GMO crop presents a contamination risk for your crop. For corn, 660 feet is considered sufficient to reliably prevent pollen drift. The distances required for alfalfa can be affected by the types of pollinating insects.
It should be 1,500 feet when alkali or leafcutter bees are stalked as pollinators, and half a mile when honeybees are stalked.
Canola is required to be a quarter mile from field mustards, including weeds.
Canola should be 660 feet from other canola.
Because these large separation distances are not always achievable, you may also need to use strategies to separate by time. There is an additional strategy that may help in the case of GMO crops with the Bt trait. If your neighbors are growing Bt corn, they are required to plant a non-Bt refuge area. One suggestion is to ask your conventional neighbors to place their refuges where they will serve as a buffer between their Bt crop and your organic crop. However, some Bt corn products now use so-called refuge in the bag and are not required to have the separate refuge areas. In corn, you can protect your crop by ensuring that your corn's pollination window does not overlap with neighboring GMO corn.
This means to plan so that your corn crop silking does not coincide with your neighbor's GMO corn pollen shed.
Pollen shed can last up to 2 weeks, while the corn silk receptiveness to pollen can be up to 2.5 weeks. What you want to avoid is exposing your corn to neighbors' GMO pollen. At a minimum, you want a week between GMO and organic corn tasseling, and ideally 3 to 4 weeks to account for unevenness in flowering times across different areas of the field. Generally, organic farmers are going to be planting later than conventional farmers, so this means you want your neighbor's corn to be finished with its pollen shedding stage before viable silks are present on your corn. You can also choose varieties with different days to flowering to help maximize this separation.
You can choose different maturities as well.
And you can monitor your fields to ensure that you are not harvesting volunteer plants that may carry transgenes, as well as monitoring your harvested crop. A variety of commercial tests are now available to detect and quantify the presence of engineered genes in crops. These can allow you to see whether your management practices are successfully preventing GMO contamination of your crops. Test strips provide an inexpensive way of screening for a particular gene that you are concerned about. Lab-based methods such as PCR and ELISA are expensive, but can quantify the amount of GMO material present and can detect the full range of commercially available modified genes. These are more often used by buyers. In addition to protecting your crops, it's important to know and protect your access to markets for those crops so that you won't lose revenue to GMO contamination.
Remember to save seed and crop test results.
Keeping records of your production practices, seed and input sources, and operations and equipment will help you to prevent contamination and identify the source of any contamination problems that may occur. Record keeping will also protect your certification. You want to be able to demonstrate that you have been following the procedures required by the NOP for permitted seed and inputs and segregation of organic and conventional products. As a preventative measure, you can collect samples of your crop prior to harvest. Sample high-risk and low-risk parts of your fields prior to harvest. You can have them tested on your own. It could help determine the source of contamination or lack thereof. If contamination is found, but only in one part of a field, it may be possible to mitigate the effects by removing the contaminated portion of your crop. Keep extra samples. You may find it useful to look for third-party verification of non-GMO status. The Non-GMO Verification Project offers a certification for seed. You can also look for seed suppliers who have taken the Safe Seed Pledge, an assurance that they will take a suite of measures to prevent GMO presence in their seed supplies. Particularly if you direct market or sell a value-added product, you may also be interested in seeking non-GMO certification for your products, which provides assurance to buyers of their non-GMO status.
The website for the Non-GMO Verification Project is nongmoproject.org.
If your crop is found to have GMO contamination, there are several steps you can take. First, request a second test to confirm that contamination has actually occurred. If it has, you may need to sell your crop as conventional, not organic or non-GMO. If your crop is food grade, consider the organic feed grade market, which may have less stringent requirements. If you suspect pollen drift was the source of the contamination, and it is due to a GMO crop that is registered as a pesticide, such as Bt corn, you can consider contacting your State Department of Agriculture about investigating the incident as a pesticide trespass. We have reached the end of our module. Thank you for watching.
This project is supported by the U.S. Department of Agriculture, National Institute of Food and Agriculture, Organic Transitions Program under Award Number 2013-5, 1106-2-1005. This project is supported by the U.S. Department of Agriculture, Agricultural Marketing Service, National Organic Program under the Transition to Organic Partnership Program, TOPP, in the Midwest region. Any opinions, findings, conclusions, or recommendations expressed in this publication are those of the authors and should not be construed to represent any official USDA or U.S. government determination or policy. The University of Minnesota shall provide equal access to and opportunity in its programs, facilities, and employment without regard to race, color, creed, religion, national origin, gender, age, marital status, familial status, disability, public assistance status, membership or activity in a local commission created for the purpose of dealing with discrimination, veteran status, sexual orientation, gender identity, or gender expression. For more information about this project, please see organicag.umn.edu, copyright 2026, Regents of the University of Minnesota. All rights reserved.